All right, welcome everyone to the EteRNA Open Group meeting. Sorry for the delay and all the technical difficulties we've been having today. So I'm Michelle. I'm a second-year grad student in the DAS Lab. You may know me on EteRNA as Who Am I? And I'm going to tell you guys a little bit about the new EteRNA Medicine Initiative that we are getting ready to start up, where we're trying to create RNA diagnostics using the tools in EteRNA. And as you guys probably have seen in the banner on the EteRNA website, our first target is going to be tuberculosis. So just to give a little bit of background on tuberculosis, it's a result of infection by a bacteria called Mycobacterium tuberculosis. And this bacteria attacks the lungs of the infected person. It's transmitted very easily by aerosol droplets in the air. So when people have TB, they develop a cough, and every time they cough, they get bacteria in the air. And then if you're exposed to these bacteria, then you also get TB. But part of the issue with TB is that you can have a latent infection, which basically means you have been infected by the bacteria, but you don't actually have the disease. So the bacteria are not proliferating inside your body. They're dormant and just sort of, not active. And then the latent infection can turn into disease at any point. And so part of the problem is differentiating between people who just have a latent infection, who can't spread it and don't have any symptoms, and people who have actual active TB. And when it's left untreated, it kills more than 50% of those infected. Basically, the bacteria eventually destroys your lungs and you get respiratory failure. And so here I'm just showing a map of estimated new TB cases in 2014, sorry. And you can see it's pretty widespread around the world, and especially concentrated in lower income countries. But in general, there's like many millions of new cases every year, and about 1.5 million people die from it every year. So it's a pretty serious and widespread problem. And in terms of diagnosing TB, there are two sort of typical assays that are used, at least in the in more developed countries, which are tuberculin skin tests and IGRAs. And many of you have probably gotten these tests before because it's pretty typical for people to get tested every 10 years. But the problem with these tests is that, as I mentioned earlier, the difference between latent and active. So these tests don't differentiate between these two forms of TB. And so you can't tell whether the patient should actually be treated. And it's also not very sensitive in HIV positive patients. So it's a big problem because a large proportion of TB patients are HIV positive because it's so prevalent in lower income areas of the world where HIV is relatively common. And so this can be a problem if it's used as a sort of general diagnostic. And then, so if you do test positive based on one of these tests, you have to determine whether you actually have active TB. And this requires chest x-rays, which are super impractical if people don't have an x-ray machine. And sputum cultures, where basically you take the mucus that is coughed up, which is called sputum, and you basically culture the bacteria inside of it, and then you can do this stain. So this image you see is a stain of some TB bacteria. And then, so after you culture the sputum, you can stain it and look under a microscope and see whether you actually have this tuberculosis bacteria. Problem with this is that it takes a super long time, and it requires you to get sputum samples, which can be relatively difficult. And so recently there was an RNA-based diagnostic developed, which is based on a technique called Realtime PCR. So basically, it tries to detect this gene called RPOB that is found in tuberculosis bacteria. And not only does it look for this gene, but it also looks for mutations in this gene, which are signs of antibiotic resistance. So another big problem with TB is that if someone's been treated with bacteria or with an antibiotic, they can develop a strain of TB that's resistant to antibiotics. And then when that spreads to others around them, and the others get treated with the same bacteria, it's no longer effective. And so when people are diagnosed, it's good to know whether the TB strain they've been affected with is antibiotic resistant already. And so this test is a pretty simple process. You have this reagent mix and you mix it with the sputum sample in this little cartridge thing, and then you put it in this machine for 2 hours, and then it gives you a result. And again, this relies on the sputum samples, which can be a problem, and it's still unreliable in HIV positive patients, but it's still a big step up in terms of being able to diagnose people relatively quickly and somewhat low cost. It's being subsidized greatly. So this method uses RNA to directly detect the bacteria. But another approach is to detect our body's response to the bacteria. So we know quite a bit about how our immune system responds once it's been attacked by TB. And the details of this figure aren't too important, but basically the point is that we can basically measure changes in RNA levels in our body that are a result of our body's response to the bacteria instead of trying to detect the bacteria directly itself. And some advantages of this are that it can be used to monitor treatment response. So if you just look for bacteria or no bacteria, that doesn't tell you about how well you're doing, how well your body is responding to treatments. This can also be blood-based. So as I mentioned earlier, many of the current treatments or current diagnostics are based on sputum, and it can be a little difficult to get the sputum sample. So it would be nice to have a blood-based diagnostic. We hope that we'll be able to create something where we can get a really rapid result, kind of like the existing RNA-based diagnostic. And also we're hoping that it's HIV invariant. So basically it'll work just as well in HIV positive and HIV negative patients. And generally we want it to be like a generalizable diagnostic. We want it to, work in all different races and all different conditions based on like the many different places that TB is found. So what we need is to find an RNA signature. So basically we want a bunch of RNAs that we think are going to change between active TB and non-TB patients. And so the way that we discover these RNAs that tell us about whether someone has TB is this multi-cohort analysis, which is developed by the Khatri lab here at Stanford. So essentially what they do is they use this microarray. So basically a microarray is a way to measure RNA levels in a sample for many, many different genes. And so what you end up with is RNA or gene expression levels for non-patients and patients, and then you can compare those and figure out which ones change the most. So they pulled some data from these public gene expression databases. one called GEO, the Gene Expression Omnibus, and one called Array Express. So these are both publicly available data sets. You can just like go online and download all this data. And so they got over 2,000 samples worth of, or 2,000 samples of microarray data from either active TB patients or controls that were from TB studies. And also, they made sure that all of the data sets they got were from blood samples, because they want to eventually do this test in blood, so they want to know about RNA expression in blood. And so they basically take the TB versus not TB and compare and see which ones change the most. So we call that differential expression. And then they use these two different measures. Oh, so I should mention that these thousands of samples were from a bunch of different cohorts from different studies. And so it's from all over the world, like different types of patients. And so we think that if we use such a diverse data set in order to learn our RNA signature, then our RNA signature will be relatively robust and generalizable. And so they use these two different measures to determine how big the effect of each gene is. And the details of these particular methods aren't too important, but it's just a way to combine the data across multiple cohorts. And so basically they end up with a list of genes, some of which are higher in TB patients and some of which are lower in TB patients. And they combine these into what they call a TB score. So basically they found these two genes, we'll call them A and B for now, which are more highly expressed in TB patients, and a gene C, which is more lowly expressed in TB patients. And so if we calculate this value, A times B, or the concentration of A times the concentration of B over the concentration of C squared, then we end up with a score that tells us about whether someone is likely to have TB or not. So here in the figure, you see on the left, healthy controls and people with latent TB, so they don't actually have the disease. And they have relatively low TB scores. And in red, you see active TB, so you can see the distribution shifts up quite a bit. And then in purple and blue, you see people that have been treated for some amount of time, and you can see that it goes down over time. So we can actually use this to measure how well someone is responding to the treatment. And so what we want to do with EteRNA is try to design an RNA molecule that can calculate this TB score. So if we put someone's blood, all the RNA from someone's blood into a tube, and we want an RNA that can tell what the value of A times B over C squared is in that tube, basically. So we're going to start with a somewhat simpler problem before we try to tackle that. And that's to just calculate a quotient, so like concentration of A over concentration of B. And so here I'm just showing a figure of what that might look like. So on the x-axis, we have the concentration of A on a log scale, and on the y-axis, we have the concentration of B, and the color represents basically the signal that we get. So it's a fraction forming MS2 in this case. And so basically what we want is the signal to be high when we have high A and low B, and the signal to be lower when we have low A and high B. And ideally what we'd have is a super sharp cutoff where if A over B is, if A over B is maybe greater than 0, then we have, we turn on, and if it's less than 0, then we turn off. And so how are we going to create an RNA that does this? So we developed this scheme.
How do you get below 0?
What do you mean? Oh, wait.
So in this last, go back one.
Yeah. Oh yeah, so these are log transformed values. So So that's how you get below 0. Okay, so the way that we're going to do this is a two-state system where we have one state where the state 1 where the RNA is bound to just B and not A, and state 2 where it's bound to just A and not B and forms the output signal. So here you can imagine that if the concentration of A goes up, we're going to get more of state 2 because more A's are able to bind to the RNA and we'll get higher signal. And if we have more B, then you'll have more RNAs in state 1 and you'll have a lower signal. So intuitively, that might sort of make sense that this represents A over B. But we can sort of show that in a more detailed way. So basically, you can write a rate of conversion between these two states. So you can imagine for state 1 to convert into state 2, you need a molecule of A and a molecule of the RNA in state 1 to basically like run into each other, and this is dependent on those two concentrations, the product of those two concentrations, and then multiplied by some constant, which is not too important right now. And the opposite is also, or the reverse rate is similar, where you want the concentration of the B molecules times the concentration of the RNA molecules in state 2. And so you end up with these two forward and backward rates. And at equilibrium, we want those two to be equal. And that's sort of what we want to be able to measure is, okay, well, let me just move on. Okay, so taking that equation at the top, and we just rearrange it a little, we get the proportion of state 1 relative to state 2. And what we really want is the proportion of RNAs that are forming MS2, because that's what we're really going to measure, is basically the amount of fluorescence. So the proportion of RNAs that are lighting up, basically. And so that's state 2, which is the one that had MS2, over the sum of both of them. And so if we plug in these equations, we end up with this equation on the right. And then we make this sort of simplifying assumption that the concentration of B is much greater than A, which is actually true in the case that we're talking about. The gene in the denominator is generally at a much higher concentration in the cell, and so, or in the blood. And so if we make this assumption, then we end up this ratio of A over B times these constants. And so this is just showing that this does indeed give us a signal that is related to the quantity A over B. And so here I'm showing the same thing that I showed before, but we, as you guys know from all of the switch experiments that you guys have worked on, we don't get a like complete binary switch like I showed earlier. It's more like a spectrum where it, gradually turns, the signal gradually turns on and off, basically. And so since we have this sort of whole concentration space that we want to explore, we can't, as you guys see in EteRNA, we usually specify a few states where we want to satisfy specific criteria. And so as you guys have probably seen in the lab puzzles recently, we've devised two different models that we can actually turn into EteRNA puzzles that you guys can actually solve more concretely. So the first one is a two-state model. And we're measuring these two states that I'm showing in these little white bubbles, where both are at the same concentration of B, but state 2 is at a higher concentration of A, and state 1 is at a lower concentration of A. And we ask that in state 1, B is bound and there's no MS2 hairpin. So it's the same as that state 1 that I showed. Let me just flash back. It's the same as this state 1 and state 2, where we have B bound in the first and A bound in the second, and also MS2 in the second. And so we set these concentrations at 2 different concentrations of A in order to cause the switching, if that makes sense. And then we also have a 3-state model where we're measuring at 3 different concentrations of A and B. And that way we can detect effects of both changes in B and changes in A. And so we ask for MS2 hairpin in this state number 2, oopsies. And when you increase the concentration of B, you turn off, and when you decrease the concentration of A, you also turn off. And so, yeah, so that forces your RNA to be sensitive to both concentrations. And so what we can actually do is use this tool called NewPack to simulate the RNA folding. So NewPack basically uses these energetic parameters that you guys know and love from EteRNA, but and then is able to calculate basically a probability for each different state of the RNA. And so this is just a simulation of a random sequence that I took from the lab puzzles. And we can see that it actually sort of approximates the behavior that we want with the sort of diagonally increasing gradient. But as you can see on the axes, the concentrations are very different than what I was showing before, which was sort of in the nanomolar range. Instead, you see that concentration of B is much higher than the concentration of A. And you'll remember that that's the simplifying assumption that we made earlier when we wanted this RNA to approximate the ratio A over B. So that gives us some hope that perhaps this will actually work as a diagnostic, but we have yet to test these, as you guys know. But in addition to having you guys design these RNAs, we're also going to have EteRNA design some sequences that we can test. And just to sort of go over how EteRNA works for those of you who don't know, basically it starts with a random sequence of all A's, just like in EteRNA. And then we pick a random base and mutate it to a random other base. And we use NewPack to predict how this RNA is going to fold in each of the different states. And based on that, we calculate a score, which is meant to represent how far we are from where we want to be. And this score is, there are 2 different scoring metrics. So the first is a base pair distance metric. So here you see the secondary structure represented in dot bracket notation. So basically the parentheses mean that those 2 bases are paired to each other, and dots mean those are unpaired. And so the top is a representation for the MS2 hairpin, and the bottom is some example for how our RNA currently folds. And basically you just compare them at each position and say, is it folding correctly at that position? And if it is, then you add 1 to the score, and if not, then you do not. And so you use that to sort of quantify how well we're doing right now. And but as you guys know, most of the time when you make a single mutation, the fold doesn't actually change. And so this score is not going to actually change. And so we want a more continuous scoring function that we can use to get a better sense of how well our designs are doing. And so we have the space pair probability score, which is basically takes each pair that we want to form, each base pair that we want to form in the final secondary structure, and adds the probabilities of those base pairs forming. And so that gives us a more continuous measurement of how well we're doing. And so we use a score to decide whether or not we're going to accept the mutation that we made. So it's proportional to this quantity that I'm showing here. But basically the general idea is that if the score got better, then you automatically accept it. And if the score got worse, then you accept it with a probability that's related to how much worse it got. And there's also this T parameter, which is sort of like a temperature. So we start at very high temperature, which means that we're more likely to go to worse solutions. And we gradually decrease the temperature, meaning that we start getting more and more stringent as time goes on. So we start accepting worse sequences less and less often. So this is represented by this little animation that you see here. So basically you start at high temperature and you sample lots of different points in sequence space. And as you gradually decrease the temperature, you start moving around a lot less, and eventually you converge to this optimal score or sequence. And here's just a quick demonstration of how it works. So you start with all A's, and you can tell it how many iterations, so how many mutations you want to make. And then gradually it changes the sequence over time and hopefully gets closer and closer to the secondary structure that we actually want. Okay, so eventually we want to be able to test these on chip. And so basically what we would really want ideally is to test this whole space and see basically all of these concentrations of A against all these concentrations of B, and be able to get a signal at every single point in this plot. But unfortunately, we can't test every single condition. So what we can do is test this sort of square of concentrations. So, and so basically you can start at some concentration of B and gradually increase the concentration of A and make a measurement over that entire line. And then once you get to a higher concentration of A, you can gradually increase the concentration of B and measure this edge and do the same for these to get this whole square of measurements. And that gives you some sense for what this whole space might look like. And what we can also do is test which oligos are bound in different states. And that will also give us a sense for whether we're actually getting this state 1 and state 2 that I showed earlier. And so lastly, eventually, we want to move towards actually calculating that TB score that I showed earlier, which is A times B over C squared. And so in order to do that, we're going to have a pretty similar setup, but instead of just one molecule bound in each state, we're going to have two molecules. So in state 1, where we're forming them as two hairpin, we have both A and B bound. And in state 2, we have two molecules of C bound, since it's C squared. And so this will be the sort of concept for the RNA that computes the full TB score. And that will be forthcoming once we've worked out all the details for the A over B puzzle. So that's basically it. I just wanted to thank the entire DOS Lab and EteRNA dev team, especially Riju, Johan, and Nando, who helped work out this A over B puzzle. To all of you who are amazing at what you do and help us design these cool RNAs, to my PhD program, and to my funding. Thanks all for tuning in, and if you guys have questions, just type them in the chat and I'll try to answer them. Is anyone posting stuff? Is anyone on the chat? Are you on chat?
There's one question from Machine Labs. Is the fluorescent detection something that can be done on site / in the field for rapid detection, like the MIT case and microscope, which is more complex than that?
Yeah, so eventually we want to move.
Can you repeat it? I don't know if
Oh, so the question was whether we will actually be able to do the fluorescence detection in the field when we're actually trying to diagnose people. And basically, our eventual goal is to move towards something that's a little bit easier, such as like paper-based diagnostics, where you can just spot a sample onto a piece of paper and the resulting color tells you whether or not you have TB, basically. And we're working with collaborators now to figure out the best way to do that.
that was the one question I see in the chat right now.
Okay.
Wait, so you had one more question before that. On the source of genetic material being measured. Let me just scroll up. Is the expression of genetic material sourced from the infecting organism, i.e. not a result of changing the person's own genetic expression due to infection?
Okay, so I think the question is whether these changes in RNA expression that we're detecting are changes from the bacteria itself or from the RNA of the host organism or the human. And the answer is that it's from the person. So basically the RNA in their blood is as a result of, or the changes in the RNA expression in their blood is as a result of their immune system responding to the TB being present in their body. So you're not actually detecting the TB bacteria itself. Yeah.
So did they test other patients that were infected by other bacteria?
Like HIV, other bacteria.
Like, how can we really say that this is a unique expression for TB and not say like a class of...
Yeah, they did test against other diseases. I didn't show that data here.
Okay.
But yeah.
That's what I mean. Okay.
So one of their control cohorts was like people with other diseases that are not TB. And
Okay.
it,
So they were like different expressions.
yeah, it's like specific to TB and works in people with HIV or not. So that's a really nice feature that is not true of most existing diagnostics.
Okay, here's a follow-up question. Does this count as epigenetics or not, if it's not permanent?
So the question was whether or not this counts as epigenetics. And so this is totally unrelated to epigenetics. So those are actual modifications to the DNA. And this is just measuring the amount of RNA in the blood, if that makes sense.
So you don't make more RNA, that's your DNA, epigenetics?
Yes. Okay, so I should say that, so epigenetics can affect RNA expression, but that's not what we're measuring here.
Maybe I can add one more question. I've seen a few comments in the chat about the puzzles being very hard. Do you have any comments or suggestions for that?
Yeah, so Johan is saying that people are commenting that the puzzles are very hard. So we're gonna try to start building like a sort of like training for the A over B puzzle. So a bunch of relatively easy or easier puzzles that use the same RNAs and use similar concepts and try to sort of train people to get them thinking in the same way that they would need to solve the A over B puzzle, and hopefully that will make it somewhat easier. And possibly at some point in the future, we might also, integrate EteRNA so it could like sort of solve some subsequence or some part of the puzzle, and then you could tweak other parts in order to improve it. But that is still in development. Nothing else?
Well, the follow-up question to the previous one was, so it's not a measure of the quantity of particular kinds of RNA, not a change in the kind of RNA expressed, just a change in the quantity of RNA expressed.
So I think it's a question about whether we're measuring changes in the types of RNA being expressed versus the amount of RNA being expressed.
Yeah, I'm not totally sure.
So we're measuring 3 different types of RNAs, which we're calling A, B, and C for now, just to make things simpler. And we're measuring the changes in the amounts of all 3 of those RNAs. Hopefully that answers the question.
Those are the questions I saw in the chat.
Okay. Well, if people have more questions about EteRNA medicine stuff in general, feel free to PM me or, in EteRNA, woo am I. And thanks for tuning in.